In this video, I will go over the mechanism of action of different bacterial toxins, starting with Shiga toxin. So the other name for Shiga toxin is verotoxin, and it's produced by both Shigella and enterohemorrhagic E. coli 0157H7. And the mechanism of action of this toxin is by blocking the 60S ribosomal RNA, as a consequence of which it will cause bloody diarrhea. And then patients may also develop hemolytic uretic syndrome, which presents with hemolytic anemia and schistocytes, thrombocytopenia, as well as the acute kidney injury. Clostridium tetani and Clostridium botulinum are working by inhibiting the SNARE protein. So SNARE protein is a protein that is required for the fusion of the vesicles in the neuronal nerve ending. So if the vesicles cannot fuse, then the neurotransmitters will not be exocytosed from those neurons. So Clostridium tetani is inhibiting the release of the inhibitory neurotransmitters like GABA and glycine, as a consequence of which these patients present with the spastic paralysis and jaw lock. On the other hand, Clostridium botulinum inhibits the release of the acetylcholine excitatory neurotransmitter, as a consequence of which these patients present with flaccid paralysis. And then I would like you to know that the infection of Clostridium tetani usually starts by inoculation of spores by stepping on rusty nails, while Clostridium botulinum, the infection can start by ingesting of the preformed toxins inside cans, so this will be foodborne, and then alternatively by ingestion of spores that is present in raw honey. Children can develop floppy baby syndrome. So in this condition, in the second condition, once inside the body, the botulinum will start to make the toxin, while with the foodborne, toxin has been previously formed. The next toxin is the toxic shock syndrome toxin of the Staphylococcus aureus, and this is a super antigen. So once there is antigen inside the body, antigen presenting cells will uptake that one, have it processed, inserted into the invariable region of the MHC2, present it on the cell surface, and then T cell receptors will recognize the antigen and induce an immune response. Now with a super antigen, they don't require to be processed by the antigen presenting cells, and they can directly interact with the invariable region of the MHC2. So there would be a vast activation of T cells in patients with the super antigen, and these patients present with fever, hypotension, as well as skin manifestations. Now, conditions that can predispose to toxic shock syndrome include the use of highly absorbent tampons, as a consequence of which the bacteria can stay there, grow, and produce the toxin. The next two bacterial toxins are produced by enterotoxigenic E. coli, or ETEC, and there are two different forms of toxin that are produced by this type of bacteria. One is the heat-labile, which will increase the cyclic AMP, as a consequence of which it increases the chloride secretion inside the gut, and thus there would be water efflux, and thus it causes watery diarrhea. While with the heat-stable toxin, there is an increased cyclic GMP, which will decrease resorption of salts. So cyclic AMP increases secretion of chloride, while cyclic GMP decreases resorption of salts, as a consequence of which water will stay inside the gut, and again there would be watery diarrhea with both cases. And the mnemonic that you can use is that as stable as a ground and as labile as an air, to know which type of toxin is associated with cyclic AMP and cyclic GMP. The next bacteria is Bacillus anthracis, which will cause a skin necrosis with an area of surrounding edema, and the surrounding edema is due to the increased cyclic AMP. So these patients present with skin death with surrounding edema. The next two conditions, Vibrio cholera and Bordetella pertussis, both of them increase cyclic AMP. But the difference is that Vibrio cholera activates a stimulating G protein, as a consequence of which it will overactivate adenylate cyclase, and then therefore there would be increased cyclic AMP, while Bordetella pertussis inhibits an inhibitory G protein, as a consequence of which there would be overactivation of adenylate cyclase, and then again there would be increased cyclic AMP. So Vibrio cholera, due to the increased secretion of chloride, presents with a rice water stool, while patients with Bordetella pertussis, due to the increased activation of the adenylate cyclase and increased concentration of cyclic AMP, they will have increased histamine sensitivity, as a consequence of which they present with a whooping cough. The next condition, Clostridium perfringens, it produces the lecithinase, also known as phospholipase C, which will break down the phospholipid lecithin. And thus these patients present with the gas, gangrene, or myonecrosis, and hemolysis. The next two bacteria, Pseudomonas and diphtheria, both of them work by inactivating the elongation factor 2, and Pseudomonas does that by producing exotoxin A, while diphtheria does that by producing an exotoxin that has been acquired from beta prophage. And then Pseudomonas causes cell death, while diphtheria presents with pharyngitis, as well as a grayish pharyngeal exudate. And here is an example of an pharyngeal exudate seen with diphtheria. Here at the back is the pseudomembrane. And then the final bacteria is Streptococcus pyogenes, which produces two different forms of toxin. One is streptolysin O, which will degrade the cell membrane, as a consequence of which it will cause beta hemolysis of the red blood cells. And then the other type of toxin is the exotoxin A, which is also a superantigen, and it will cause toxic shock syndrome due to the increased release of interferon gamma and IL-2. And that concludes our discussion.